Let's talk more about the prokaryotic cell. Every cell, prokaryotic or eukaryotic, has four must-haves: a plasma membrane, a cytoplasm, which is the jelly stuff on the inside, genetic material, DNA and RNA, and ribosomes, where the protein magic happens. Most prokaryotes come in three main shapes. They can be looking like spheres, or like rods, or like spirals. You can imagine different spaghetti shape, for example. So all cells have those four main components. They have a plasma membrane surrounding them. They have a cytoplasm, that's the jelly stuff. Then they have genetic material, the DNA and the RNA, and they have ribosomes, which are the protein factories. And see, down in here, and I gotta get out of the way a little bit, or we can see the different shapes. We have cocci, they look like spheres, right here. We have bacilli, they look like rods. And then we have spirilla, and they look like spirals. They're like the different shape of spaghetti. Well, maybe the round one is more like a matzo ball. What do you think? Prokaryotes, the bacteria and archaea, don't do the whole membrane-bound organelle thing. So they don't have a nucleus. Instead, they have a single circular chromosome chilling around in the nucleoid region. See here? You can see here. Let me get out of the way again. Look at that. A nucleoid region. That's this area here where we see the chromosomes in. And see, here's the chromosomes. You can see that in here. Most also have a cell wall outside the plasma membrane for extra protection, shape, and general tough guy vibes. This is where then the bacteria and archaea differ significantly, because they have differences in the cell membrane and the cell wall, but we'll talk about that in a minute. Some species have a capsule. Think of it like a cozy jacket that helps them stick out onto surfaces and avoid drying out. Others have flagella, which are little tails that whip and move them around. Even others have pili, tiny hair-like structures to latch onto surfaces or even other bacteria. We'll call that conjugation, but more of that in a minute as well. Plasmids are bonus rings of DNA that can pass around special traits like antibiotic resistance, for example. We can see all of them here, the cell membrane and cell wall. You see right here, the darker green and then the lighter green. This picture also shows the extra features. A capsule, right here, the capsule, the lighter green on the outside. A flagellum, which is the word for a single one. Here you see the flagellum. Flagella is a multiple, and there is multiples. And then we even have some pili, the small extension helping them latch onto things. See right here, these are the small extensions, the pili. Bacteria and archaea both are prokaryotes, but they differ in how the cell membrane and wall are built. This is a sign of an ancient separation in their lineages. Some archaeal membranes are lipid monolayers instead of phospholipid bilayers, which is how we mostly understand the cell membrane structure at this point. Bacterial cell walls have peptidoglycan, which is kind of a mesh of sugar and peptides, which are amino acid chains. Bacteria can be gram-positive or gram-negative. The positive ones have many layers of peptidoglycans, making their walls thicker than the gram-negative ones. They only have one or a few layers and are thinner. Instead, they have an additional membrane on their outside. See here, we have gram-positive bacteria, and we can see the multi-layer of the peptidoglycan wall. And then over here, we have the gram-negative bacteria. We can see the single layer of peptidoglycans, but we have that outer membrane that we can see here instead. Archaea don't use peptidoglycan at all. They've got their own unique cell wall recipes. One type uses pseudopeptidoglycans. The other three types identified The other three types identified contain polysaccharides, which are a sugary glucose type structure, glycoproteins, which are sugars attached to protein, and a specific cell surface layer of proteins known as the S layer. Variety is the spice of life. Prokaryotes usually reproduce asexually via binary fission. They replicate their circular DNA loop, attach the copies to a membrane, and then split. Boom, two identical clones. No mitosis here, just a straightforward copy and split approach. While binary fission doesn't shuffle the genes much, prokaryotes have a few sneaky ways actually of mixing them up. In transformation, they pick up stray DNA from the environment. If it's from a nasty pathogen, that can turn a harmless bacteria into a troublemaker. In transduction, viruses vary DNA they pick up from one bacteria to another. We call those bacteriophages. And thirdly, in conjugation, DNA is directly transferred by a pilus from one cell to another. Think of it like tiny little straws-like structures that are creating channels for genetic goodies to move from one cell to another. It's often plasmids that get traded this way. So binary fission is asexual reproduction of prokaryotes where a parent cell divides into two identical daughter cells. Binary fission does not shuffle the genes. Prokaryotes create genetic diversity by transformation, when a cell picks up DNA from the environment and incorporates it into their genome. Transduction, when the genetic material is transferred from one bacterium to another via a virus, a bacteriophage we call that. And conjugation, when genetic material is transferred from one bacterium to another through direct contact. Because many prokaryotes can divide in minutes and quickly swap genes, they adapt to new challenges, like antibiotics, lightning fast. Remember that next time you wonder why antibiotic resistance is such a big deal. Prokaryotes fill a bunch of jobs on Earth. They recycle nutrients like nitrogen and carbon when they break down dead stuff, for example. Some even live inside other organisms. Yep, we have them in our own guts. Probiotics, anyone? There are those. Different prokaryotes use different sources of energy to assemble macromolecules from smaller ones. Phototrophs use sunlight for energy, kind of like little solar panels give us electricity. And chemotrophs, on the other hand, you get their energy from different chemical compounds. So, when we look at the sources of energy prokaryotes utilize to assemble macromolecules from smaller molecules, we look at phototrophs, which obtain their energy through sunlight, and the other group is the chemotrophs, which get their energy through chemical compounds. So there you have it. Biofilm, cell walls, reproduction, and more. Next time you're scrubbing the sink and reading about antibiotic resistance, give not to these microscopic rascals. They are everywhere. Let's go and look a little bit deeper in the ones that make us sick.